The overall goal of this indirect coculture assay is to investigate the impact of astrocytes on the development of neurons.
Our laboratory is interested in the role of neuron-glia interactions. It is believed that astrocytes contribute to the formation of the synapses, the connecting structures of the central nervous system. In order to study their role, we have designed an in vitro model where we can combine primary astrocytes and primary embryonic neurons in separate compartments. The compartments are connected by a permeable membrane, which allows for the analysis of exchanges between both cell types. Using this approach, we have been able to monitor synapse formation over periods of up to 4 weeks. In addition, it is possible to investigate the individual roles of astrocytes on the one hand and neurons on the other hand using this assay. And finally, we can also investigate in more detail the secretome of our cell cultures, which contains molecules that mediate the reciprocal influence of both cell compartments in that system.
This method can provide insights into the interactions between neurons and astrocytes. Also, it can be applied to other model organisms, such as the cells from rat.
Generally, individuals new to this method will struggle because experience is required to obtain the optimal trituration of hippocampal tissue after dissection.